Well, let us start today. One of the most interesting topic in this biotechnology principle and process. That is the end of this topic. That how the recombinant cell we have produced and its culture is maintained by genetic engineering technology. अब उस recombinant cell से हमारा product of interest जो है specific what you call protein or vaccine or hormone. Now that how we are going to produce that we are going to see today, and for that we need a device. That device is called fermenter or bioreactor. It is being called fermenter because most of the product are produced by the process of fermentation, and it is also called bioreactor more precisely because the nutrient medium through which we are producing the product by using specific microorganism, they are living system. Hence the reaction is being caused by these microbes or the cells or their enzyme. That's why it is also called bioreactor. Now how it is made up of? Basically it is a cylindrical vat-like structure. It is made up of basically stainless steel. Remember, when the size is in thousands to lakhs of liter, it is made up of stainless steel. But when for experimentation purpose or research purpose it is built up to 10 liter, 20 liter, it is made up of Pyrex glass for what you call convenient in the laboratory. Okay. In general, how this functions. So how the product is being produced. Before going deep into that, let us understand its what you call architecture. Basically it is a cylindrical vat like structure made up of stainless steel. Okay, now what does it consist of? See here, it is a motor which runs on electric current, and it is basically the agitation system of this bioreactor. What this motor or agitator system does, it rotates all the medium plus microorganism present inside this cylinder. Okay, then these impeller blades, okay, they causes turbulence while agitation. Because of that, maximum mixing of the nutrient plus the oxygen to be done inside this vat. Okay, now during production condition may changes, acidity increases, alkalinity increases. To check on that, this control system is present here, a buffer system is here to maintain the proper pH of the this production medium. Okay, it do have this sterile air is being passed from here. This entire workspace can be divided into two spaces. Okay, one, this one down, that is the working volume of this bioreactor. Okay, this one down is the working volume of this bioreactor, and above this is the head space. Okay, so it has two volume, workspace volume and head space volume. This workspace volume is almost 70% of any bioreactor, and almost 20 to 30% of this head space is being for what you call during production the foam is generated, and that foam is being broken here in this head space. Okay, and to sterilize it different methods are applied. Basically here through this vat what you call jacket, sterilized steam is being passed. Okay, so hot steam is being passed, that causes sterilization of inconvenient or contaminant present in this bioreactor. Now these are two types of this bioreactor. One, this is stirred tank, other is sparged tank. Basically both are same, only the difference is that in case of sparged tank bioreactor, this oxygen supply is more. Because of this air bubbles are present here, they use more surface area for microorganism and more oxygen for particle their growth and multiplication inside the medium. Overall both are same. One major difference between them is that this sparge tank bioreactor has a special sparger through which the air is being passed, sterile air is being passed, and this air increases the surface area inside in the form of bubbles. So you can see these bubbles increases the surface area for the activity of microbes present inside this bioreactor. Now when we are using this bioreactor, two types of cultures are being done. One is continuous, another one is batch. जब किसी फर्मेंटर के अंदर कंटिन्यूस प्रोडक्ट प्रोड्यूस करना होता है, तो एक बार प्रोडक्ट प्रोड्यूस होने के बाद जो वेस्ट है या फिर स्लरी है उसको रिमूव करके फ्रेश मीडियम ऐड किया जाता है, एंड इन दिस वे दैट बायोरिएक्टर इज फंक्शनिंग कंटिन्यूअसली. In case of batch culture, if one batch production is done, it is stopped, product is purified, and then the second batch start after the initial beginning. So in this way, two types of cultures are being done, continuous and batch culture. Now come to ये foreign gene product obtain करते कैसे हैं? This is just the device. Two methods are there, processes are there. One is USP, another one is DSP. USP is the upstream processing. From the recombinant cell production with desired gene, the product which we want, to the type of medium it require. उस मीडियम का ऑप्टिमाइजेशन, उसकी ग्रोथ कंडीशन का ऑप्टिमाइजेशन एंड इट्स प्रोडक्शन कंडीशन का ऑप्टिमाइजेशन जो होता है, all these processes comes under upstream processing. Then once we get the desired microorganism, the desired nutrient medium, desired temperature or pH condition or physiological condition सब optimize होने के बाद this start functioning, and once we produce this product inside this optimum condition of the bioreactor, finally product produce होने के बाद उसको isolate करके purify करके marketing करने की जो process होती है, that is DSP or downstream processing. So downstream processing starts after production of the product inside the bioreactor. Now how this DSP is being done? Simple. First of all, the DSP requires one principle, that जो product produce हुआ है, whether it is extracellular or intracellular. If the product is extracellular, it will get accumulated or dissolved inside this liquid present inside this bioreactor. If it is intracellular, then definitely we need all those insular cell debris. By breaking them, we can take our product. So that must be remember whether the product is extracellular or intracellular. Okay, fine. Now what are the general methods? सबसे पहले हमको insolubles को remove करना पड़ेगा. How we are going to do this? By filtration. This slurry or this what you call the remnant present inside this bioreactor is passed through the filter, and then the filter is being taken. Depending upon the product is extra or intracellular, the product is being selected from that liquid or from insoluble debris. Flocculation. In this method, these insolubles are allowed to clump together to form the flock, and finally those flocks are being taken out. Sedimentation. It is actually the insoluble particles present inside the liquids are allowed to sediment. In the same way, precipitation is also emergence of any insoluble present in the liquid to the bottom, that is precipitation. Centrifugation. Okay, here this is also one of the insoluble removing method. यहाँ पे centrifugation के अंदर centrifugal force apply किया जाता है, and by applying that centrifugal force, the molecules settle at the bottom depending upon their size and mass. So these are simply the removal of insolubles, that is first step in DSP. Then the product which is being separated from these insolubles has to be isolated. अब उसको कैसे remove करेंगे? There are these methods for product isolation, that is solvent extraction, absorption, ultrafiltration. Okay, so solvent extraction कैसे करते हैं? Suppose this is one liquid in which our product is there after filtration. Okay, so this product after filtration in the liquid is one medium. What we'll do in that liquid medium, we will add one more medium in which our product is more soluble, and by mixing these two liquid, our product will get dissolved into the immiscible another solvent added, and that solvent is being separated. That is called solvent extraction. In case of absorption, after this insoluble removal, this absorbent bed is what you call bed is being kept up, on which the molecules or the product get retained on the surface of the absorbent. In case of ultrafiltration, it is simply the membrane filtration. Okay, here semi-permeable membrane is being used. We know that our product has slightly more molecular size than the water. So this water liquid plus our product is passed through the semi-permeable membrane. The product is retained because of its size, and the water flows through the membrane. In this way, our product can be isolated once it is separated from the insolubles in the medium. Then we have to purify the product, because the product purification is a key for the quality maintenance. So how we are going to purify? Simply the method applied for product purifications are chromatography and crystallization. In case of chromatography, this product after purification is being allowed in a solvent system, and that solvent system run depending upon its retention rate inside that solvent. और अनदर मेथड है क्रिस्टलाइजेशन. यहाँ पे क्या करते हैं, उस प्रोडक्ट को एक अल्टीमेट क्रिस्टल फॉर्म में आने के लिए मजबूर करते हैं, जैसे फ्रीजिंग, एंड इट गेट क्रिस्टलाइज एंड फाइनली दैट प्रोडक्ट इज प्यूरिफाइड. वंस द प्रोडक्ट प्यूरिफिकेशन इज डन, हम क्या करेंगे, उसको फॉर्मुलेशन करेंगे, बिकॉज उसकी क्वालिटी खराब नहीं होनी चाहिए. उसकी सेल्फ लाइफ बढ़ाने के लिए हम उसमें प्रिजर्वेटिव्स ऐड करेंगे. दैट इज फॉर्मुलेशन बाय प्रिजर्वेटिव. फॉर एग्जांपल, मिथिल आइसोथायोअज़ोलिनोन इज अ प्रिजर्वेटिव that is antimicrobial, antibacterial, antifungal, methyl isothiazolidine. Okay, next is quality testing. Once all this insoluble is removed, product is isolated, product is purified, preservative is added, it is tested for its quality, whether it is good than the current product present in the market. Then certification is being done from the government certifying authorities, and then it is released into the market with a particular label and price tag. That is how we obtain the foreign gene product of interest by recombinant DNA technology. So it is simply a sequential step to understand what is the theme of the topic, how this functions, how it is made up of, why it is called bioreactor, what is its architecture, what is the major difference, then what are the specialities, then what is USP, what is medium, what is optimum condition, what is DSP, and finally what are the different steps of the DSP. This can be asked for separate question. What are the removal of insoluble methods, or product isolation methods, or product purification methods, or any example of preservative. In this way we obtain the foreign gene product by recombinant DNA technology, and I wish you understood this simply. So thank you, thanks for watching.